Okay, this is a video for grade 12 biology, and I'm going to be using various images from the internet and taking some from the Nelson Biology 12 textbook. I want to go over transcription and translation just a little bit, but I want to focus on the lac and Tripoperon, positive gene control, eukaryotic gene control, and then there's going to be an assignment on mutations from the textbook. Okay, so the first thing we've got here is a little overview of transcription and translation. And I know the diagrams are a little bit small to see, but recall that we've got DNA here in the nucleus. And through the process of transcription over here, RNA is produced. And if we focus on messenger RNA travels out through a nuclear pore into the cytoplasm here. So here we have our mRNA. And mRNA interacts with the ribosome. Of course, the ribosome has a couple of subunits to it. Recall it's got a small and a large subunit. Now you can see that better over here in this diagram, the small and the large subunit. But if we go back to this diagram, you can see that tRNA, transfer RNA, another type of RNA which is produced in the nucleus, carries amino acids. So amino acids are carried by tRNA, and they come over here and they attach, and the anticodon of the tRNA binds to the codon of mRNA, and that specifies whether or not the amino acid is the correct one. The ribosome moves one codon at a time down the mRNA, and the polypeptide chain is created. So this is the process of translation over here, whereas over here we had the process of transcription. Now, if we look at the larger diagram over here, we can see another view of the same thing. You can see over here DNA is unzipping, so there's the strand that is being used to build the mRNA. So here is messenger RNA. Messenger RNA then leaves the nucleus into the cytoplasm, and in the first step of translation, it interacts with a small subunit of a ribosome. Here you can see a transfer RNA coming in, and the large subunit of the ribosome attaching, and that's the initiation phase. And then during the elongation phase, you can see over here, first of all, the tRNAs are carrying the amino acids, so various shapes representing the different amino acids that are possible on transfer RNA. The tRNA anticodon here binds with the codon, and you can sort of see that the spots on the large subunit of the ribosome where the tRNAs come in, and the amino acids are transferred over, building a long polypeptide chain. Now, in this example, you're going to see that the amino acid is transferred from the right side to the left side, but it's still always from the P site to the A site. So check the diagram that I gave you to see that better. So we have transcription going on in the nucleus and translation going on down here. And you've also looked at reading the genetic code. So you can look up in that table, the mRNA table, looking at the codon AAG, you would find that it codes for a particular amino acid. Okay, so what we really want to do here, though, is we want to focus on, hang on. Now this is called negative control, and we're going to be looking at the Lactose Operon or the Lactose Operon. So an operon is a cluster of genes, and here we can see what we've referred to as the structural genes here, the beta-galactosidase genes. We also have an operator and a promoter, and we've got a repressor protein or a LAC-L protein as well. Now, in the absence of lactose, or when lactose is low, we don't have lactose, which is symbolized here by this circle. And so the repressor protein will bind to a region on the DNA of a prokaryote, let's say bacteria in this case, right? It will bind to a region called the operator. So when the LAC-L or repressor protein binds to the operator, it overlaps, and you can see that here in this color diagram. It overlaps a region called the promoter. The promoter is the site that RNA polymerase has to bind to right there in order to move down the beta-galactosidase genes or the structural genes and transcribe messenger RNA. So in the absence of lactose, when the repressor binds to the operator, overlapping the promoter, RNA polymerase cannot bind, and we don't want it to bind and transcribe the genes to make mRNA because we don't need those enzymes. It's senseless to make the enzymes that break down lactose when lactose is in such low concentration or not present at all. When lactose is present, when it is in high concentration, here's the lactose. It binds to the repressor and causes a change in conformation or change in shape. Because of that, the repressor cannot properly bind to the operator region. So that would be O right there, operator. That would be promoter. So because the repressor has been changed in its shape due to the binding of lactose, it can't bind to the operator. RNA polymerase then can bind to its special region on the DNA called the promoter, and then it will move down the structural genes, causing the production of this thing here, and that represents the mRNA. That, of course, will then be translated, and the enzymes beta-galactosidase, permease, and transacetylase will be produced, and they will start to break down the lactose. You can read about the specific functions of these in the Nelson textbook as well. As these enzymes function and act on the lactose starts to decrease in concentration, decreased concentration of lactose, put an L in there, and as lactose becomes unavailable, we go back to this state where the repressor protein is not bound anymore, and therefore its shape is the normal shape, binds to the operator, overlaps the promoter. RNA polymerase cannot get in. All right, so lactose is called an activator or an inducer because it turns the operon on. In other words, the presence of lactose over here, the presence of this stuff, changing the shape of the repressor causes this to be turned on and RNA polymerase can work. So lactose is an activator or an inducer in this case. Now I want to talk next about negative control, and this is a new operon that we never really talked about. This is the Tripoperon or the tryptophan operon. Now tryptophan is an amino acid, and here we can see the structure of this amino acid with the amino group, the carboxyl group, and the R group is showing here. Okay, and there'd be another hydrogen off of a main carbon as well. But that's the structure of tryptophan, an amino acid. So in order to produce tryptophan when it is in low concentration, and it's an essential amino acid that the bacteria need, so they need to make this amino acid to make various proteins. In order to produce tryptophan, 5 genes are going to be involved. Each of these genes needs to be transcribed so that messenger RNA can be produced, which is then going to be translated, and tryptophan, TRP, is going to be produced. So this is the case where there is low tryptophan and tryptophan needs to be created. In this scenario, all right, in this case, there is a repressor protein just as in the lac operon, and it is produced in its inactive form. So you can see that it's already bent in this case. So in low tryptophan, the repressor is produced in an inactive form. It then cannot bind to the operator of the Tripoperon. This is a different operon, all right? It's called the Tripoperon. The repressor cannot bind. RNA polymerase then is free to bind to its promoter and transcribe these genes, which will produce the mRNA, which when translated will create the enzymes necessary to produce this stuff, to make this. Now, we're not breaking it down like the lac operon. We're making tryptophan. All right, so low tryptophan, inactive repressor, RNA polymerase works. Now, when tryptophan is high, we don't need to transcribe these genes to make more tryptophan. So we need a way to turn the Tripoperon off in this case. Now in this case, tryptophan, similar to the way lactose works, tryptophan here binds to the repressor, this guy here, changing its shape so that it does fit into the operator here. When the repressor is active, because tryptophan has bonded to it, it fits into the operator, and while they don't show it very well, it overlaps the promoter. Again, this is just the reverse of the lactose operon. It's got a lot of similarities and a few differences. So when tryptophan is present, it binds to the repressor, changing its shape, but now the repressor is active and able to bind to the operator. RNA polymerase therefore cannot get into the promoter, so we do not have any transcription of these genes, and so we don't produce tryptophan. So that is called negative control. Now, there is positive control as well. And positive control is when the binding of a regulatory molecule, in this case, this thing here, the binding of a regulatory molecule actually promotes transcription, whereas in the previous case, the binding of a regulatory molecule, the repressor, let me get rid of that, the repressor shut off the operon. But in this case, the binding of a regulatory molecule turns on the operon. So we're going to look at the lac operon again, and there's something called CAP or CRP. So either CAP or CRP. We say CAP, catabolite activator protein or regulator protein. All right. In this image, they use CRP. And when lactose is present, we want to produce, well, the bacteria wants to produce the enzymes to degrade the lactose, right? So in the presence of lactose, you need to produce those enzymes so that you can degrade the lactose and produce energy. So when lactose is present, all right, and when glucose is absent, in other words, the glucose is being used up because that's easier to use the monosaccharide than the disaccharide. Lactose is present. We haven't broken it down yet. So the bacteria have been living off the glucose, using it up. And as they use up glucose, they, of course, are going to produce ATP. And as the ATP is used in various ways, you're going to produce ADP. But as that is used in various ways, you're also going to produce AMP, adenosine monophosphate, which can be converted to cyclic AMP just with a small conversion. Now, you've seen this molecule as a second messenger for hormones, and it's a similar idea. All right, so we have a molecule called cyclic AMP produced when glucose is low. So low glucose, using up the glucose, high cyclic AMP, this inverse relationship. Cyclic AMP then attaches to this CAP or CRP, which is inactive. The binding of cyclic AMP to the CAP causes it to become active. And when it's active, it can bind to the DNA as well, and it assists the action of RNA polymerase, perhaps by touching it or pushing it. Or think of it like this way. If RNA polymerase had a positive charge here, and if this CAP had a positive charge, as it bonded, there would be some repulsion that would sort of push the RNA polymerase down. So think of it like that. It assists or it helps. Down here, I've talked about helping. It helps the RNA polymerase to bind and to transcribe those lac operon genes. So lactose is present. We need to make those enzymes. Using up glucose so far. All right, so I'm going to switch over to lactose. Cyclic AMP is high. Therefore, it binds to the CAP protein, which binds to the DNA, to the operon, assisting RNA polymerase. Now, when lactose is present, again, but glucose hasn't been used very much, all right? There's plentiful glucose. In that case, there's not going to be as much cyclic AMP because it hasn't been converted into cyclic AMP. Therefore, we don't see cyclic AMP here. That CAP protein stays inactive, and then it doesn't bind to the lac operon. Therefore, RNA polymerase doesn't function as effectively. Now, I know in a previous example, we did not even see this CAP. We just saw, hey, if the lactose is present, as it is in this circle here, it's going to cause a change in shape of the repressor. The repressor can't bind to the operator. Therefore, RNA polymerase can bind to its promoter and transcribe the genes, right? That's what we saw before. And that's fine, but the truth is there's a little bit more to it. This CAP is necessary to help the RNA polymerase work. So in the presence of lactose, even though the repressor has changed shape and won't bind to the operator, we need this CAP here. All right, so we want lactose to be present. Yes, of course, that'll bend the repressor, as you can see in this case. But we also want glucose to be used up so that the bacteria have a reason to switch from glucose over to lactose. If there's plenty of glucose, don't worry about lactose, right? But if the glucose is running low, the inverse of that will occur, and cyclic AMP will be high, which will bind to the cap protein activated, and that will help transcription. This is called positive control because the binding of a regulatory molecule, in this case the catabolite regulator protein, stimulates the operon, all right? Turns the operon on in this case so that it works. Whereas without the binding, the operon is essentially off, similar to if you had a normal repressor protein binding to the operator. So it's another level of complexity, admittedly. So you need to understand the lac operon, how it works in its on and off state, as we did before, and then add this other level to it, which is the cap, understanding the concentrations of lactose and glucose. Now, I want to suggest to you that you consider writing some of these points down. All right, so teacher can pause this. And I also want to suggest to you that you look for other diagrams on the internet. This exact diagram is available. Perhaps you could print it out and put it in your notes, and that will help you to learn this material. Okay, let's move on. Now, I know that this image is not going to be very clear. This comes from your textbook, and it is the 4 levels of control of gene expression in eukaryotic cells. This comes from the homework that I asked you to do, the printout sheet on eukaryotic gene control. There are 4 levels of control: transcriptional control, post-transcriptional, after transcription, translational, and post-translational, after translation. Now, we are not going to be looking at any operons in grade 12 bio for eukaryotic cells. We're just going to look at these sort of 4 processes. Now, there's a diagram over here, this just comes from the internet as well, that shows DNA undergoes replication, and then transcription occurs, producing messenger RNA, really all the types of RNA, but let's focus on mRNA. Translation occurs, and that produces a polypeptide or a protein. And this is where those 4 stages would occur. We've got transcriptional controls that control the rate at which DNA is transcribed. Okay, so here it says it regulates which genes are transcribed or controls the rate at which transcription occurs. That's transcriptional control. Now we have the mRNA, and then there's going to be post-transcriptional modification. All right, after transcription, the mRNA is going to be modified in certain ways. We'll talk about that in a moment. And then during translation, this can be controlled as well. So translational control controls how often and how rapidly the mRNA transcripts will be translated into proteins. This control affects the length of time it takes for mRNA to be activated and the speed at which cytoplasmic enzymes destroy mRNA. Because once the mRNA has been translated, you've got this mRNA strand here, you've already created your protein, that mRNA can either be translated again or it's going to be chopped up by ribonuclease, and its ribonucleotides are going to be recycled. All right, so all of this can be controlled. Post-translational modification is once the polypeptide or the protein is produced, it's going to be modified. Now, it says here, before many proteins become functional, they must pass through the cell membrane. A number of control mechanisms affect the rate at which a protein becomes active and the time that it remains functional, including the addition of various chemical groups. So just imagine, to make it a little bit simpler, often ribosomes are attached to the endoplasmic reticulum. So there's the ER and there's a ribosome. So R for ribosome, ER for endoplasmic reticulum. Now let me switch to another color here. Let's say I go to blue. As the protein is produced, it's often inserted into the endoplasmic reticulum, and this is what will transport it to the Golgi for packaging and then released from the cell. There are many enzymes functioning right here in the ER that will modify the protein. You can put on various functional groups. You can attach polypeptides together. So there are various places where proteins or polypeptides are modified after translation. So this is post-translational modification. I want to go back, though, and talk about post-transcriptional modification, and that deals with this diagram here that I asked you to either draw or to photocopy. I can't quite remember which one it was. This is called a primary transcript. This is a piece of messenger RNA, mRNA, and it is not modified yet. The DNA has various regions called exons and introns. Introns are intervening regions that are sort of like junk stuff. Kind of like when I record a song or whatever and I want to cut out some of my vocal or something like that, or you're editing a movie or even an image in Photoshop and you're going to get rid of some of the stuff that you don't want. You're going to crop it and put things together. Exons are the expressed regions. But I mean, if I have a piece of DNA right here and we just copy it, straight transcription, you're going to get some extra pieces in there that aren't really useful. So these have to be cut out. In addition to that, a cap is going to be placed on the 5' end and a tail called a poly A tail. This is with adenine or adenosine nucleotides at the very end. This cap is going to help the messenger RNA bind to the ribosome. So remember, you've got the small subunit here, you've got the large subunit of a ribosome, and the mRNA is going to come in and it's going to attach, and you've got the various sites on the thing, and then you've got your codons along here. Well, this cap is going to be right here. It's going to help the mRNA bind to the ribosome. But as the mRNA travels through the cytoplasm, there are enzymes that are chewing at it all the time, sort of trying to break it down. And this poly A tail is going to help prevent the mRNA from being degraded. In other words, those things are going to be attacked first right here and chewed away so that by the time it gets to the ribosome, hopefully none of the good stuff has been broken down. So this is the poly A tail, this is the 5' cap. This is part of post-transcriptional modification. And I also mentioned these introns here. So there's an enzyme called a spliceosome, and this enzyme is going to cut out those introns right there and splice together the exons. So the good parts are going to be joined together, the bad stuff is going to be removed, and here we have a mature mRNA transcript, or a final mRNA transcript, just with exons. Now again, this is just a piece of messenger RNA with all of the bases here, all right? Just imagine, though, that the crappy ones have been removed, the wasted stuff, and only the good stuff is here. This will be translated. These are the bases that will bind to the tRNA anticodons, 3 at a time, right, to make up codons. And you've got the 5' cap and the poly A tail. This is post-transcriptional modification, and I wanted to go into it a little bit deeper than the other ones because we have the removal of the introns, the addition of the 5' cap, and the poly A tail. So that's 3 things there to be familiar with. At this point, I'm going to stop the video, and we can freeze it at this point, and you can quickly copy this down and copy this down. It'll take you about 5 minutes to do that. And here is a screen capture of the part in the textbook, in the Nelson Biology 12 textbook, that I want you to read through. It's this one here, and then on to the next couple of pages. It's section 5.6. It deals with mutations. And what I'd like you to do is to define these terms. Now, we're going to talk about these terms in class, and I'll give you some examples. So you go ahead, define the terms. I mean, it's always a good idea to skim through this, read through this. Most of the terms are right here. Define these terms. And then on the next page, you've got figure 1, and it shows a graphic of how these mutations work. I want you to study that figure after you've defined these and consider photocopying it and putting it in your notes. All right, so that you've got a good reference, and we can move on with all these definitions and that figure, and of course any of the previous figures of I've given you Lac Operon, or I think I had you photocopy it, but you're going to need Lac Operon, Tripoperon, and I really want to suggest that you find a nice image to show you this positive control with the cap or the CRP. All right, so I'm going to move back here. I'm going to leave it there. Hope that helps. See you soon.